Equisetum introduction. It is the only living genus of class Phenopsida having about 30 species. The most common and worldwide distributed species is Equisetum avens. It is commonly known as horsetail. Habitat. It is worldwide in distribution but most common in temperate regions. It generally grows in moist places. General structure. The plant body is sporophyte. It is composed of rhizome, aerial branches, scale leaves and roots. Rhizome. Plant body has horizontal underground rhizome. Rhizome gives off erect aerial branches. Rhizome and aerial branches have nodes and internodes. Internodes have ridges and furrows. Lateral branches arise from the nodes. Some buds produce short branches called tubers. Tubers give rise to new plants' vegetative reproduction. Leaves. Each node has a whorl of small scale leaves. These leaves form sheathing at the base of node. Leaves perform little photosynthesis. Roots. Roots arise in whorls at the nodes of the rhizome. Aerial branches. The aerial branches are green and perform photosynthesis. The aerial branches are differentiated into nodes and internodes. Some species of Equisetum have two types of aerial branches. Fertile branches. Fertile branches are short and brownish in color. They are without lateral branches and are produced in spring. Each fertile branch produces a cone or strobilus at the apex. Sterile branches. The sterile branches are green and have numerous lateral branches. These branches persist throughout the year. Internal structure. Internal structure of the stem, aerial branches or rhizome. Internally the stem is differentiated into following structures. Epidermis. It is the outermost layer and consists of a single layer of cells. Cell wall of these cells is highly silicified. Stomata are present in the epidermis of aerial branches but absent in the rhizome. Cortex. Cortex is present between epidermis and endodermis. Cortex has long canals called vallicular canals. Cortex is composed of parenchymatous cells. There are large bands of sclerenchyma in the peripheral portion of the cortex. These sclerenchyma bands form the main supporting tissue. The cortical cells of aerial branches contain chloroplasts. They are called chlorenchyma. They are the main photosynthetic tissue of the plant. These chlorenchyma cells are absent in rhizome. Endodermis. Endodermis is present in a two cortex. Endodermis is formed of a single layer. These cells have the characteristic Casparian strips or bands on the radial walls. Pericycle. Pericycle is present in a to the endodermis. It consists of a single layer of parenchymatous cells. Stellae. Parenchymatous cells form the ground tissue of the stellae. Pith is present in the center. In the primary aerial branch, this pith has central canal which is absent in lateral branches and in rhizome. Stellae type is siphonostele. The vascular bundles are arranged in a ring around pith. Each vascular bundle is collateral. In this case, xylem is inner and phloem is outer. Mature vascular bundles have a cavity called carinal canal. Internal structure of the root. Each mature root has a single layered epidermis. Cortex is composed of parenchymatous cells. Two layers of endodermis are present below the cortex. Cells of the inner layer of endodermis give rise to the secondary branches. A definite pericycle is absent in roots. Stellae is present in the center. It is without pith. Internal structure of leaf. Each leaf has a single vein. Vein is surrounded by the endodermis. Its vascular bundle is collateral. There is no carinal canal. Life cycle of Equisetum. Sporangia strobilus asexual reproduction. Equisetum as homosporous spores are produced within the sporangium. The sporangia are borne on the sporangiophores which are aggregated into a compact structure called strobilus or cone. The cones or strobili are produced at the apex of fertile branches. Each cone has an elongated central axis. Sporophylls or sporangiophores are attached on it. A ring-like outgrowth annulus is present at the base of the axis. The number of sporangia on each sporophyll varies from 5 to 10. Development of the sporangium. A single cell initiates the development of each sporangium. It divides into an inner and an outer cell. The inner cell further divides to produce the sporogenous tissue or archosporium. Outer cell takes part in formation the sporangial wall. Wall of the sporangium consists of several layers of cells. The innermost layer is tapetum. 1/3 archosporial cells enlarge and become spore mother cells. The remaining cells disintegrate to form mucilaginous liquid. This liquid provides the nourishment to the developing spores. Each spore mother cell divides meiotically to form 4 spores. Wall of spore becomes 4-layered. The outermost layer epispore splits to form 4 bands. These bands separate from the spore wall on drying. These bands are called alaters. These alaters help in the splitting of the sporangial wall and dispersal of spores. Germination of spore and formation of prothallus. The spore germinates on suitable substratum. It divides into 2 unequal cells. Smaller cell forms the first rhizoid. The larger cell divides to produces prothallus. Peripheral cells of prothallus are meristematic. They divide to increase the size of prothallus and produce gametophyte. Sex organs are produced on the upper side of gametophyte. Sex organs, sexual reproduction. Sex organs are produced at the margin of the prothallus. But later they are embedded in the thallus. Antheridium shocked face. Each antheridium is produced from a superficial cell that divides to produce outer jacket initial and inner androgonial initial. The jacket initial divides to produce wall of the antheridium. The wall has a triangular opercular cell at the top. The androgonial initial divides to produce mass of androgonial cells. Each androgonial cell divides to produce 2 androcytes or antherozoid mother cells. Each androcyte is changed into an antherozoid. The mature antherozoids come out by the lifting of the opercular cell of wall. Archegonium shocked face. Archegonium also develops from a superficial cell that divides to produce an upper neck initial and a lower central cell. The neck initial divides to produce a neck. The central cell divides transversely to produce an upper neck canal initial and lower ventral cell. The neck canal initial produces a row of 2 or 3 neck canal cells. The ventral cell divides to produce a large egg or oosphere at the base and a smaller ventral canal cell. Fertilization. The ventral canal cell and the neck canal cell of mature archegonia disintegrate to form passage for the anthrozoids. Several anthrozoids enter the archegonia. But only one of them fuses with the egg and forms zygote. Development of the sporophyte. The oospore divides mitotically to form embryo that later divides into an upper epibasal cell and a lower hypobasal cell. The hypobasal cell divides to produce a foot and the first root. The epibasal cell divides to form an apical cell and adjacent cell. The apical cell produces the first branch, primary branch. The primary branch produces 1 or more secondary branches at its base. Secondary branches develop their own adventitious roots. One secondary branch grows horizontally into the soil and forms the rhizome that gives rise to the vertical aerial branches. The adjacent cells produce the first whorl of 3 scale leaves. The primary branch develops adventitious roots at its base.